Okay. Thank you very much to the organizers for inviting me. I was an undergraduate student here at the Material Science Beamline 20 years ago. And it's great to see that the ESRF is still that very exciting, bubbly scientific community it was back then. So I'm speaking in this session about methods, so I thought I was obliged to show you something about the methods development we've done, and then I'd like to focus on examples that our lab has happened to work on in Alzheimer's disease. So I'd like to start with this slide, which is just from Richard's paper in 1995 already, where he promised us, based on theoretical considerations, that we should be able to build de novo atomic models in particles, and these numbers were later adjusted even downwards, and for molecules that are only about 100,000 Dalton big. Now, for many years, people thought that Richard was rather the optimist, and that's perhaps explained by the cloud of black points here, which are all structures from the 21st century. So these are between the year 2000 and the year 2012 in the electron microscopy data bank. And I plotted molecular weight versus the resolution obtained. And you can see that most of these are in the kind of what we now call the blob range, where individual protein domains are one blob. And it's great to study quaternary structure, but not great to study structural biology at the atomic level. So some reconstructions would then go to kind of sub-nanometer resolution, where you can see alpha helices and so on. Things become a bit more defined. Now, if you would go down all the way outside into the outside the building and very large viruses, things were kind of moving towards atomic. But here on the small side, there was nothing close to where you would be able to build an atomic model in the map. And then suddenly, 2013, 2014, we got structures coming out which were around 3.5, 3 angstroms resolution, and we could suddenly build these models. And that's still ongoing, of course, and Sriram and Jean kind of gave a nice introduction as well. And we can see that now the number of EM structures is expanding very rapidly, and I guess that's one of the main reasons we're all here today as well. So that does raise the question, what happened, what caused this sudden rapid increase after the many years of evolution also that Sriram mentioned. So I think the main reason there is the development of hardware for detectors. So we got better detectors that lose fewer electrons in the detection process, and there's now currently 3 companies selling these things, and all 3 are better than what we used before on photographic film. And this is from a review from Richard's group a few years ago. Now, along with the development of better detectors have also been another development, and that's in better image processing. That's closer to my own heart. And there was part of this is because these new detectors are much faster, so you can record movies rather than still images. And then if your sample moves while you put the electron beam on it, you can correct for it in these movies. That will give you higher resolution. So not only are the images better, we can also correct for some of the artifacts. So the same amount of images will give a lot more information than we could on photographic film, for example. Now, another part of better image processing is the possibility to classify out distinct conformational states. And that's where cryo-electron microscopy, although we measure images of tens or hundreds or even millions of individual molecules, each image is just that. It's a single molecule imaging technique. So provided we're capable of separating images of distinct conformational states in the computer, we could then reconstruct multiple states from mixtures. And that I think is one of the most promising aspects of cryo-EM. Okay, so that's then closest to my own heart. We develop in my group this software package called Regularized Likelihood Optimization, or RELION in short. And I won't go into the mathematical details of the approaches. The entire reconstruction problem is expressed in an empirical Bayesian framework. And what that means for the user is that very critical signal-to-noise parameters are estimated iteratively from the data themselves. So this is kind of this Bayesian inference-like phenomena, and thereby the need for expert users to tune all these parameters is strongly reduced. So it's relatively easy for users who do not really know what's going on to still get excellent high-resolution reconstruction. And then also this framework turns out to be very powerful at doing three-dimensional classification, separating out these images of these three-dimensional conformational states that are all slightly different. And I'll give you a few examples later on. Now, recently we engaged with a collaboration with the group of Erik Lindahl, who's at the SciLife lab in Stockholm, and these are Björn and Dari, his very talented students. And they've implemented this GPU acceleration of RELION, and basically, and this is an example of data from 3RAM of the beta-gal, which he also showed. Basically, now we can process these very large data sets faster than we can acquire the data. So that kind of, before perhaps image processing was very computationally intensive, you'd need very big clusters and it could become a bottleneck. Now for a machine that costs about 5,000 to 10,000 euros, and your nephews and cousins would be very happy to play very nice three-dimensional games on it. But we can make use of the same type of hardware to process data just in a few days, whereas probably this data took much longer to collect. So that removes at least this big bottleneck of processing the data. Storing all the terabytes of data that come out of your microscope will still remain a problem, I guess, also here. So then this aspects of being able to classify out different conformational state kind of touches upon a major problem of structural biology. If we do somehow have a mixture of molecules because our purification process wasn't good enough yet, then typically in structural biology, whether you do NMR or crystallography or previously cryo-EM as well, would be to run even more columns and try and separate it all out, and then hopefully end up with separate Appendorf tubes where you have a homogeneous sample prep. Now, to some extent, and some people get overly excited by this, but we can now surpass this, and we could directly image this mixture, and then we can let the computer figure it all out and get more than one structure out of a single experiment straight away. So we call that in silico purification of proteins. Okay, one example is actually from Alexei, who's here today, and this is when he was a postdoc in Venki Ramakrishnan's lab at the LMB, and he had purified mitochondrial ribosomes from yeast, and apparently, I'm not an expert at all, but apparently when you do this, you have these mitochondrial membrane preps, cytoplasmic ribosomes stick to the cytoplasmic side of these membranes, and he pulls them then later out the ribosomes, and he gets a mixture of mitochondrial and cytoplasmic ribosomes. So at some point he asked us, it will take 6 months more to do more columns and so on, and then Xiaochen Bai, a postdoc in my group, imaged this mixture, and he took images of 100,000 individual ribosome particles, and then just used RELION to get rid of about 30% contamination of cytoplasmic ribosomes. So these are they're quite different from the mitochondrial ones, and it was relatively easy for the software to do this in silico purification. And then we went on to calculate a 3.2 overall angstrom resolution map of the large subunit of this mitochondrial ribosome. Now, that then posed another problem, a new one for us at least, where we had been used to rotating blobs around in blob rotating software. Suddenly, we had to use X-ray crystallography software, so this is just a screenshot from Qt, and we were staring at 3 or 4 megadaltons full of near atomic resolution map, and we had to build an atomic model into this. And it's really thanks to these people, Garib, who writes RefMac, and Paul, who writes Qt, who are now also at the LMB, that we could go from that empty map to a fully geometrically restrained atomic model inside this map. So that still took Alexei and Alan and several other people, I think, 6 months of work in front of the graphics station. So there's still room for automation and so on there for those of you in the audience who are into computer program development. But I think that was that was a great breakthrough. So just as an example of what is possible now, I want to briefly mention two projects that have gone on in my lab, one not so recent and the other one very recent. So they're both from Alzheimer's disease, and this neurodegenerative disorder is defined by, if you look in the brains of Alzheimer's patients, when they die, they have extracellular amyloid beta deposits and intracellular inclusions of a protein called tau. And both of these, the aggregates seem to be like amyloid, so kind of forming all these extended beta sheet fibrils. Now, the first example is related to the amyloid deposits, because it's based on that's about the structure of human gamma secretase. And this is a collaboration with Yigong Shi at Tsinghua University in China, who sent us purified protein. And then Xiaochen Bai, the same postdoc in my lab who imaged the mitochondrial ribosomes, he made cryo-EM grids, imaged it, and solved the structure. So gamma secretase cuts many membrane-bound proteins, many of them, as long as they have a single transmembrane helix, many proteins can be a substrate for gamma secretase, not just one of it, and that relates gamma secretase to cancer, but the amyloid precursor protein, which is then cut to form these amyloid beta peptides, is another one, and that relates it to Alzheimer's disease. Now, gamma secretase is a membrane protein complex, so there's 4 different proteins, all of which have at least one transmembrane helix, which come all together in the membrane. Then there's one large ectodomain, which is also heavily glycosylated. So that makes it a nightmare for structural biology in general, I guess. People have tried to crystallize this for decades without having any success. Also for cryo-EM, when we did this with a total mass of 170 kilodaltons of protein, unknown asymmetrical complex was a challenge. And that's what we like to do in my lab. We like to work on challenging cryo-EM samples together in collaboration with biological groups to, in that way, drive the methods forward. So just another illustration that this was challenging for cryo-EM. There had been several structures of gamma secretase by EM in the blob era, and all of them looked disconcertingly different. So we were pleased to, with the development of new methods for taking into account movement and radiation damage in these movies, which were developed for this project, to finally end up with a 3.4 angstrom structure. So this is the actual reconstructed density, and this is the model that was built in de novo. Now, I don't have any time to go into the details of the gamma secretase story, but I did wanted to point out one aspect is that when we looked at the active site, which is here in the blue subunit, that was actually the most disordered part of the protein complex, where we could see less well-defined densities. And that was kind of interesting because we have this very promiscuous protease that cuts many different substrates. And when it cuts amyloid precursor protein, it can do so also in a sloppy way, because it can lead to either very short or not so short amyloid beta peptides. And then particularly the longer ones have a stronger tendency to accumulate in the brain of these Alzheimer's patients. So we have a promiscuous, very sloppy protein, which has a relatively disordered active site that we thought was interesting from a structural biology point of view. And we thought, well, if we do all these classifications, and we tried hard to do classifications on this to make this better, but it didn't really work. So we had to come up with a new classification method, again, where the actual biological project drives the method forward, where we subtract part of the density and focus classification on a small part where we know things are going on. And that way we were capable of separating out 3 distinct conformational states by focusing our classifications only on the CN subunit. And at the end, the differences between the 3 states is just rotations and order or disorder of just a few alpha helices. So I think that's very exciting that now we can use these classification algorithms to look at dynamics of protein molecules at the secondary structure level. And this is just one example, and I'm sure you'll see more examples today and both tomorrow. Okay. Now, the main example I actually wanted to show you is on the structure of these tau proteins, the other protein that forms the intracellular tangles. And this is a collaboration with Michel Goddard, who's also group leader at the LMB, and this work is mainly the work of Anthony Fitzpatrick, who is a postdoc in my lab. So Tau is a protein that occurs in 6 different isoforms in the brain, so you have splicing variants, and most notably there's this, in the full-length tau, the longest isoform is 441 amino acids, and there's 4 repeats, which are called the microtubule binding repeats. And it's, in its happy, healthy life, the tau protein is thought to bind to microtubules and stabilize these, and thereby contribute somehow to neuronal health, although we don't really know the details. Now, because of the splicing of exon 10, you can then have 4 repeat tau, where the second repeat is present, or this can be spliced out, and then you have only 3 repeat tau. And then you get 6 isoforms by splicing in some regions here at the N-terminus. Now, Alzheimer's disease is only one in a very long list of so-called tauopathies. So these are all neurodegenerative diseases, and they're neuropathologically very different, and also the symptoms that the people have with these diseases is very different. So in some disease you might lose speech, in the other you might lose inhibition or memory, like in Alzheimer's disease. Now, in all the other tauopathies, there is no amyloid beta inclusions. That only happens in Alzheimer's disease. So these only have tau accumulations. Now, as I said, the neuropathology of these different tau diseases changes. So I don't know what these pictures mean, but the neuropathology tells me they're all very different. And interestingly, for a structural biology audience and a structural biologist as myself, if you now look at the inclusions at these fibrils of amyloids in the different diseases, you can see that different isoforms of tau accumulate differently in the disease. So there's Pick's disease, where you only have accumulation of 3 repeat isoforms, and there's other diseases where you only have 4 repeat isoforms forming fibrils. And Alzheimer's disease is one example where you have just a combination of both 3 and 4 repeat isoforms accumulating in these inclusions. Now, if you then look at these filaments with negative stain electron microscopy, and these are pictures by Tony Crowther already in the 80s, I think, you can see that also ultrastructurally, these filaments look very different. So this is from Alzheimer's disease. This is called a paired helical filament and a straight filament. There's 2 different types of filaments. I'll come back to that. And then this is from a 4 repeat only tau inclusion and a 3 repeat only tau inclusion. These filaments look very different. One other thing about tau is that people have proposed, and the last word isn't out yet, but that it might be infectious. So if you have these amyloid-like conformations, aggregates of tau, they might act like, perhaps like a prion does, so that this unhealthy conformation of tau is capable somehow of convincing healthily ordered tau in the brain to accumulate and grow these fibrils. And the ideas of that are from several directions. There seems to be some kind of spreading of tau during disease, so it starts in one part of the brain and then seems to spread with subsequent stages of the disease. And also Michel did these experiments before, where they have a mouse overexpressing human tau, and they then inject brain extract from patients with some of these tauopathy. Then the mouse will develop neuropathology and also symptoms of that tauopathy. And if you inject it with a different type of tauopathy, that mouse will develop different types of neuropathology and symptoms. So that all then raises the question, does tau perhaps behave like a prion? And would there then be kind of different strains, different conformations, which are all unhealthy and infectious, related to different diseases? So I think that's where structural biology comes in quite nice. It's a understanding that at an atomic level is where structural biology could come in handy. Minneapolis sent us samples of a brain of a 74-year-old American lady who had died of Alzheimer's disease. And we homogenized the cortex regions and did a Sarkozy insoluble fraction. And then you can spin that down. And then you can do, this is a negative stain picture. You can see these typical paired helical filaments and straight filaments, which have been associated with Alzheimer's disease. Now, then if you want to do an atomic structure of these are amyloid-like structures. And here this is already a result, but these are 2D class averages of the PHFs and the SFs. You can see that along the helical axis, these fibrils are very, very smooth. Only apart from this kind of striation, these kind of horizontal lines, which are actually 4.7 angstroms apart. So that's the distance between two beta strands in a beta sheet. And that makes that they're Fourier transformed. There's a lot of information in the XY plane of these filaments, but along that, along the helical axis, there's no information at all until you get to 4.7 angstroms. So if you want to align individual projection images of amyloid fibrils, you have to do that straight at 4.7 angstroms. It's not like a single particle which has information scattered throughout the Fourier transform, and you can kind of gradually grow in resolution. Amyloids have therefore been notoriously difficult to solve. And again, that's why we like working on these kind of samples, because Xiaoda He, who was a student, PhD student in my lab, had been working on extending our software to do helical reconstruction. And he expressed several of these statistical priors that could help and make this all more stable refinement. So we used his software to then calculate now to 3.4, 3.5 angstroms resolution atomic structures of both the paired helical and the straight filaments from this Alzheimer's disease brain. And along the z-axis, you can see beautiful separation now of these beta strands in the very long beta helix, which basically forms these filaments. Now, if you look at the paired helical filament, now through the XY plane, you can see it's made up of two C-shaped light protofilaments who come together in a symmetrical interface here. So there's actually helical symmetry relates one C with the other one. And of this 441 amino acids in the full-length protein of the longest isoform, only number 306 to 378 forms this structured core. So there's another 305 amino acids and another 70 odd amino acids on the other side, which flop around this structured core, and they form what people have called the fuzzy coat. So there's a whole range of disorder around these ordered cores. Now the straight filament has, surprisingly to us, the exact same structures made up of the exact same amino acids, again this kind of double C-shaped structure, but now they pack in a markedly asymmetrical manner, and these are no longer related by helical symmetry with each other. Both structures more or less rotate 1 degree with every 4.7 angstrom rise, which would be now perpendicular to the plane. So the kind of crossover distance thereby remains similar. Now you can overlay the C's of the two types of structures, and then you can see that they're very similar. And here I've made, this is amino acid 1 to 441 of the longest isoform, and I've put it forms 8 beta strands within the C-shaped protofilament, and it starts actually at 306, which is the beginning of this third microtubule binding repeat. And then the structured core it goes all the way through to the fourth repeat and even into the C-terminus up until residue 378. So surprisingly, despite the very high sequence similarity between all 4 repeats, really, the repeat 3 would form this part of the structure, and repeat 4 forms a very different structure. So it's not always the case where very homologous sequences adopt very similar structures, although I'll come back to that a bit later on. Now, if you look along the helical axis, these structures are not completely flat, so they actually meander up and down a bit, and up to 10 angstroms difference. And this is a schematic representation of the structure, which makes it easier to understand it. So it starts here. There's the 8 beta strands I mentioned before. Now, this part of the structure is the typical kind of cross-beta packing, where you have anti-parallel beta sheets which pack against each other. Those are quite kind of permissive in what you have in terms of amino acids sequence. You can pack either a charged or hydrophobic residues all together, as long as you don't have too many opposing charges. And then we have glycines, which make a turn here. And then you have another stretch of kind of cross-beta packing that follows here. And cross-beta packing has been traditionally thought of as very common in amyloid structures. Now, the tip of the C is markedly different from that, and it's called the so-called beta helix structure, where you have, again, very important glycine here to make a very sharp turn. And then you can only have kind of hydrophobic residues inside the tip of the C, and you need these alternating charges on the outside. If you only want one message of what does this structure tell us, it's actually that these Cs, the disordered core structure, start at amino acid 306 and end at amino acid 378. That's very relevant, for example, for pharmaceutical industry. I was at GlaxoSmithKline a while ago, and they were doing, as were many people in the field, they used these so-called K18 and K19 constructs. And because nobody had expected that the actual ordered core would extend beyond the microtubule binding repeat regions, they stop all their constructs at 372. So I don't know what kind of structures they're forming in their in vitro assays, but they're very unlikely to be the exact ones from Alzheimer's disease. So those are, I think they should at least let it run until 378, and it might be good to have a look under the microscope what these filaments actually look like. Now, this beta helix that we have that forms the tip of the C is very similar to a beta solenoid structure, which was solved with solid-state NMR of the yeast prion protein. And this beta solenoid part of the yeast prion protein is the part of the yeast prion that's responsible for its infectious behavior. That does not mean that this part of the tau structure is responsible for its protective behavior, but as you all know, structural biology is just very good at formulating hypotheses, which can then be tested by other means. Okay. Now then, almost wrapping up, the protofilament interface, we have these two very different filament-like structures. One is paired helical, one is straight, yet the protofilament is the same. So why and how are these different? We don't really know why they're different, but we can look at the interface. The symmetrical interface in the PHF is actually a very stable interface. It has lots of cross-protofilament hydrogen bonds. So we think these two protofilaments are kept together quite stably. And the straight filament protofilament interface is rather different. It comes closer together here with P312 on one side and S324 on the other side, but here might be some kind of hydrophobic exclusion or so involved in the pack, but there doesn't seem to be very strong forces keeping it together. What we do see is 4 ordered lysines, and they all point towards this extra blob of density that we don't really know what it is, and that blob of density is actually, you could see it here already in the picture that I showed you before, the same 2 lysines in the PHF on both of the protofilaments also point towards a blob of extra density. So there's something that coordinates with the positive charges on the lysine molecules, and we didn't really know what it was. And in the straight filament, they then become responsible for keeping the filament somehow folds back on itself, and then the negative charges provided by the glutamates in the EFE might be involved in coordinating those 4 lysines which come together to this extra type of density, somehow forming the interface. That again is a hypothesis. But that brings us to our model of how tau filaments now form. We show that the structured core starts at the beginning of R3, comprises all of R4 and goes then all the way into the C-terminus further than people had thought. Now, that raises the question, that kind of answers the question, why can we have a mixture of 3 and 4 repeat tau isoforms in Alzheimer's disease filaments? That is because where the 3 and the 4 repeat isoforms are different is in the presence or absence of the second repeat, which is not part of the structured core. So this is part of the fuzzy code, and you just have randomly flopping things. And then we just think you get a random incorporation of all 6 isoforms all on top of each other in the direction of the helical axis. In the other direction, in the XY plane, we think that the EFE motif at the N-terminus here with the blue dot might be involved in bringing straight filament protofilaments, the protofilaments together in the straight filaments, whereas the same kind of interaction might still happen in the paired helical filaments, although there they would not be involved in keeping the filaments together. Now, going back to this question, how can we have these different tauopathies with very ultrastructurally different types of filaments and different incorporation of only 3 or 4 repeat tau or mixtures? One hypothesis, and again it's only a hypothesis at this point, is that given the sequence similarity between the 4 microtubule binding repeats, we can, notably all these glycines which forms these corners in the structures of the C are very much conserved. So it would be possible to have C-like structures made up of other of these microtubule binding repeat. And if somehow your structured core incorporates the second one, then you would only be able to grow 4 repeat tau filaments. Or if you would have R1 to R3 core, because R2 was spliced out, then that would only be able to incorporate 3 repeat tau isoforms. So whether that's the molecular basis of differences between these tauopathies is something that will be the subject of future work. So my time is up. I come to my conclusions. New detectors and software have made possible this recent sprint in resolution in cryo-EM. I'm very excited that we can now classify secondary structure motions by 3D classification. And I think the exciting thing of being able to solve amyloid reconstructions that come straight from the brains of deceased patients opens up a whole range of new possibilities in studying neurodegenerative diseases. Just to remind you, Xiaochen Bai did the gamma secretase structure, Anthony did the tau structure. I thank you very much for your attention.
So, 2 questions.
That's absolutely beautiful. So this was from one patient, these structures. Yeah. Do you expect patient variability in
Good
structures?
Question. So we have since done patient number 2 and patient number 3 as well, and they all look the same. So we've looked at now at 3 Alzheimer's disease patients. We've repeated the experiment, and they all look the same.
One more question. Yeah.
Scheres, it's me.
Yeah.
Hi. Do you think this extra density you see with the lysine, associated with the lysine, could it be due to post-translational modifications? Or?
I don't know. It could be any polyanionic molecule kind of helping to keep those things together. It could, yeah. We'll have to see.
Okay, I think if there's no more questions, we're a little bit pressed for time, so we should move on. Thank you very much for a wonderful talk.